my talk is going to sort of give sort of lots of vignettes about drug development in colorectal cancer, with the idea that there's going to be a lot of jargon in here, and there's really no way to go through and define everything. But actually, what I want you to start seeing is what you're going to see at these review meetings and places that you go. Because the slides I'll show are very similar in terms of, you know, what kind of research is being done, what kinds of terms do we talk about. And so I thought it would be good just to sort of get it out there so that you start thinking in that mindset. And so I think most of you have probably seen this. It's kind of interesting. So this is where my career started. And I'm a phase 1. I sort of focus on early drug development. And it was actually irinotecan that we tested in phase 1. And you know, back then that was a big breakthrough. 5-FU was all we had. And the reason I always bring this up is for when patients are asking about phase 1 trials, then it's really those examples for that particular patient would be, if it works, then you actually have access to it early on in your disease, if it works. But anyway, so more recently, you guys probably need to start looking up what's out there, because there's some new drugs. TAS-102, ramucirumab, which is, they always give us ever-increasing complex names. But I think now we're out to 30-plus months for survival for patients with metastatic disease, which is really just the beginning. So I always want to start with, what do we actually have now? And so basically, many of you know we have sort of the chemotherapy stuff that's pretty active in terms of shrinking cancers. We have the EGFR targeted things that are for the patients that have wild type KRAS, which is that mutation. And then we have the VEGF. Who knows what VEGF is? So basically, any of you that got Avastin is targeting the VEGF pathway. It stands for vascular endothelial growth factor. You know, you're going to probably forget that, but just think about blood vessels. So the idea is that it's inhibiting a process of making blood vessels that are needed to grow the tumor. So that's what they do. And then, as you can see here, we have all this alphabet soup of combining this chemo with that biological agent. And so then in terms of adjuvant therapy, which is something I would love to see advocates work on, is right now the gold standard is FOLFOX, and that oxaliplatin part is a problem. And I really, I mean, I'm saying it, you have felt it, but I have patients that have permanent neuropathy, and we stopped early. So I think drug developers need to be working on a substitute. You know, you guys, please get out there, because it's interesting how few oncologists really bring this up at national meetings, and it's huge for those of us that see the patients. So this is just giving you sort of, again, this is the kind of stuff you're going to see, which is, these are basically showing you what a receptor looks like. Those little things up there we call ligands. This is like the cell membrane, and then this is on the inside. But again, you know, I'm not trying to give you a biology lesson, but just start getting used to this. So this, in yellow, are all the drugs we have that target this VEGF angiogenesis pathway. And so one of the things that frustrates me is, if you look at drug development, we made a lot of progress for a few years, and now we just keep going back to the same thing. More EGFR, more VEGF, VEGF and angiogenesis. This is the new one, and okay, what's the difference? It's just targeting a different part than Avastin targets. So that is not enough. I mean, we're not going to push the envelope if that's all we do. This was just a study design. But this is what I want to show, because you're going to see this a lot. These are basically a survival curve. And these unfortunate little ticks here are when the patients pass away. And what you're looking at is, over time, there's sort of a test for this, that we consider it significant if you can like put your finger between it. But look at that, we're talking 6.4 weeks. Now, I never,
What's the blue versus yellow?
So basically we're looking at the test arm and then the control. So you're looking at, like, let's give standard plus something, standard plus placebo. And so the good thing is it's higher. You know, the yellow one's higher than the blue one. But you need to get used to how subtle these differences are. So the difference was 6.4 weeks. It's about a month and a half, and that's, you know, that's a frustrating result for something that is sort of the next big drug approved. This I just wanted to say, TAS 102, and I'm trying to give you a flavor of where drug development is. You're going to hear a lot of these words that have to do with the names that they get from the pharmaceutical company, and I forget, it's called something VIST.
Lonsurf.
Or Lonsurf, and then there's something else anyway. Lonsurf is TAS 102. Okay, so how is it different from 5-FU? Because again, now what we're doing is coming up with more chemo that's like the other chemo. It's basically 5-FU-like drug plus something that inhibits the breakdown of that 5-FU. So it's a combination, but it's really not that different. It's basically sort of a designer 5-FU. And again, this is what you see in terms of, so there's generally two things we look at, one being overall survival. That's what we consider the gold standard, like we want people to live longer. But we also measure from the time they went on study when that scan gets worse. And so that's basically the progression-free survival, meaning when they progress, okay? So you can see here again, the yellow line's bigger than that. But I think this study was actually based on survival. And so you need to always understand the studies are designed to where they're looking at either survival as really the main endpoint or this progression question. And again, you can see there are some differences there, and it was about, yeah, two months. So that's the new drug that came out. So what have we learned? So I think one of the things that we're really trying to figure out in colorectal cancer, and you guys hear about lung cancer every day, right? We heard about, you know, CML and Gleevec. You know, we have lots of targeted agents and other diseases. Well, what we've learned in CRC is that we did have a targeted set of agents, but we really didn't spend a lot of time figuring out who was going to respond. And it turns out we treated a lot of people with KRAS mutations that derive no benefit or even a decrement. The other thing that's been interesting is we keep trying to improve by adding two sort of targeted agents together. We haven't gotten anywhere with that. These two don't work well together. And then this is basically where we basically saw more toxicity. And then I guess you all remember the BRAF story. You've seen the pictures of that guy with melanoma. Well, I mean, the most obvious next question was, we're going to give that drug to BRAF mutant colorectal cancer. Didn't work. Now, from a development standpoint, and again, this is something that you guys would think about, talk to people, because if you become engaged in clinical research advocacy, you want to start thinking about what are the important questions here. And you've lived them, so you would have a lot of insight into that, right? But the question is, where can we actually have impact? I think everywhere, as long as it gets better. But some of the things that are happening in drug development is that the pharmaceutical companies, which we partner with, it's easier to just test it right when you've been diagnosed with metastatic disease. That's the easiest scenario. And so what's happening is everything is going there, and everybody is competing with one another for that same, what we call first-line metastatic therapy. And so I've just listed some here that we're interested in. You've no doubt heard about the fact that we do more liver-directed therapy. Well, many of us don't know what to do after that, because it's not really adjuvant therapy. You know, what is it we're giving at that point? Can we give a biologic after we've removed the liver tumor, but we're a little worried that we left some cells behind, so we want to continue with the biological. And you know, this chemotherapy issue is a big one, because what we would like to do is to have a patient come in, shrink that tumor with chemo, but then not have to continue to treat them and pound them with chemo forever. So the idea would be to have biologics that you could give as maintenance therapy. We're very interested in adjuvant therapy, and I think, you know, one of the things to understand as we get into immunology, which I'm not going to give you a lecture on, but you're hearing a lot about these breakthroughs with immunotherapy. Well, in colorectal cancer, in a particular population, which is that microsatellite unstable, there are shrinkage. You know, patients do get disease regression. You know, it's been in the press. But you'll actually meet Rodney Barefoot, who basically has been on study 2.5 years with immunotherapy, and his disease hasn't gone away, but he's doing everything he wants to do. So, I mean, at the end of the day, we don't want to just stop development because we don't see shrinkage. And then there's a lot of other things we're interested in, like can we give something to basically make radiation work better?
I see you speaking of the ablative procedures, so that can be used for colon cancer in terms of the liver?
Yeah. I mean, we tend to use directed radiotherapy, stereotactic body radiotherapy, rather than that, but certainly it's still an option for liver. So anyway, you know, basically this is where we are, and I'm just going to show you, go quickly through sort of what's out there. And again, every institution that you go to is going to have their favorite handful of things that they're working on. But I want you to get a sense of how that looks in terms of where are things happening, and if this is something that you want to get into in terms of advocacy, starting to dig a little bit deeper into what's going on. And then Match will be presented later today. So one of the things that's happening is that all of that genetic sequencing that was done of many, many tumors led to the Cancer Genome Atlas project in colorectal cancer. So what you'll see is that a lot of these things here, I don't expect you to know what they are, but we are identifying the dominant pathways. Because we actually think in colorectal cancer, it's probably not as simple as you have this mutation, you get this. You have this mutation, you get that. Why? Well, in some respects, if you look at lung cancer, that was pretty much a very rapidly fatal disease until recently. And so that tumor probably developed what we call a driver mutation that just drove that cancer. And if you target that, then it slows it down or shrinks it. But colorectal cancer has been there longer, and so we think it probably picks up other things that are going on, so it's not quite, there's no one dominant oncogene that's driving it in terms of something that we can target for therapy. So one of, you know, what you'll see as treatment goes further is in colorectal cancer, because you're going to hear a lot about this mutation, the MATCH study, but keep in mind we're a little skeptical that what we need to be doing is probably some of these, all these things representing like different pathways on a road, that we need to target more than one. And so I'm not going to go too much in detail, but who knows what KRAS is? Okay, good. So KRAS targets that RAS pathway, and so here's a drug that basically targets that pathway called a MEK inhibitor. And so what you're looking at here is a study that was done, again, this is sort of that same kind of curve, but because they're all together means that there was no benefit. And so that was the case of trying to target KRAS with a MEK inhibitor. But one of the things that we've been interested in, and Chris Liu, who some of you may have met sometime, I don't know.
He did the webinar.
Yeah.
Yes.
Oh,
Yeah, so Chris is great. And so we're just working on ways to get that to work better. And so one of the things that he did was some animal studies. And again, I don't want you to worry about understanding exactly what this is, but you do need to start thinking about these animal curves, because you're going to see a lot of that if you're on a studies section. And that would be that you're always going to see, these are basically mouse tumors, and these are mouse tumors from patients, okay? So I call them xeno patients, so like little mouse patients, but they have actual human tumors. And so what we've done is we've treated some with what we call vehicle, just like control, and we've treated some that get this drug or that drug, and then this was the combination. So what are we looking at? We're looking at how they grow, just like you would on a CT scan. But we're measuring them because we can measure in mice. And so what you see is this thing that is basically the placebo just continues to grow, right? That's what it should do, because we didn't give it anything. But then we gave these other two drugs, including one of the MEK inhibitors, really nothing. But if you put them together, what a striking result that is. So this is what you want to see. You know, as a patient, if you're participating in a trial, and as an advocate, if you're advocating for robust clinical research coming out of the lab, then you want to see a flat line or getting smaller. So this would be considered positive. And then what we did here is we just wanted to go ask if the tumors were bigger, would it still work? And so basically, this was the very first clinical trial. It's ongoing. It's very focused on treatment of colorectal cancer. And it's that same combination that we treated in the mice. And it's now throughout all of those 10 NCI phase 1 centers. And so we're at the verge of enrolling sort of the expanded cohort. Andy, where am I on time? Because I can.
If you want to go another 5 to 7 minutes, does that work?
Yeah.
Okay.
So I'm not going to go over all the slides in detail, but again, going back to our pathway, the Wnt pathway, this is what Steve Leong was talking about with regards to stem cells. And this is the kind of slide you're going to see sometimes when people are saying, oh, I've got this new phase 1 trial, you know, what's going on, what's the data? And so this is basically what we did was a phase 1 study. Now, do you guys understand the concept of stem cells? Yeah. So, I mean, they're interesting. We don't know whether targeting stem cells per se is going to be extremely effective, but we've always known that in the crypts of the bowel that there are stem cells. And so the idea is that those stem cells are driving resistance to therapy. And so that's really what this pathway is doing. And so one of the exciting things is, again, in phase 1, we're often just looking to see that patients seem to have no progression while they're on study, with relatively few side effects. So that's sort of the types of things that you'll see presented. And this was basically another stem cell target. And this is interesting because, I won't show that, but again, so what do we mean when we show this, right? What do you think? So this is pre, this is post, patients on a Notch1 antibody.
It doesn't look like it changed.
Right, is that good or bad?
It didn't get bigger.
Right. And this is 9th line therapy. So these are the little glimpses. It's not where we want to be, but these are the glimpses of activity we want to see in phase 1, similar to Rodney that you'll meet, because I'm not going to discount being, you know, having a patient that's been on something for 2.5 years, does not have symptoms from his metastatic disease, and the drug has no side effects.
The tumor changed shape, is that significance at all?
No, that's just related. You know, CAT scans are funny because they go like this.
Okay.
And so based on how you breathe, it's going to change.
Got it.
Yeah, good question though. And then this is just, again, saying, how would we really benefit people? And that is, we need to have a better way to select patients. And so what Wells Messersmith's lab did is just looked at whether or not they could use a particular biomarker. And so again, this is where these patient models come up. Each one of these is a different patient-derived xenograft model. And what we like about that is that this is much more like looking at colorectal cancer tumors in patients across a population, rather than having the similarity of cell lines. Okay. Next 3 minutes on immunotherapy. Not going to tell you all the basics, but you guys, anyone in this field that wants to go, if you're going to be promoting research, should go get some review of immunotherapy, because I had to. I'm not an immunologist, but it's going to be interesting. And I think for people involved in prevention, I think it's going to be very interesting, because one of the reasons we think that older patients get cancer is related to their immune system really failing, you know? And so I wouldn't be surprised if years from now we identify high-risk patients in terms of their sort of immune system, and the immune therapy, for example, that Rodney's on, you know, doesn't have side effects. So why wouldn't you want to intervene to make a patient more responsive to a potentially fatal tumor? So that's really what this is about. Do you guys understand what those PD-1, PD-L1s, because I have a one-sentence thing for it. Okay, so just the way it's working is it's releasing a brake that has been put on an important part of your immune system. So it's really interesting. It's different than what people tried for years, which was just vaccines. Vaccines tried to stimulate, you know, like you're sitting there and you're like, okay, I want my immune system to attack this tumor. This is releasing a brake that is inherent in many cancers. So it's very interesting, and there's many ways that it's being looked at. And this is just showing that you can see that, so what we're showing here is basically the size of the tumors over time and looking at a biomarker, but mainly take home here is when you look below the line, that means it's actually shrinking. Obviously, this means it's getting bigger, and then you have some patients that have stable disease. And I think one of those is probably Rodney. And my point there being that we need to make sure that we're focusing on drugs in colorectal cancer that provide longevity and quality, and really focus on that, and not necessarily have other rigid things that need to happen. Oh, I do want you to see a waterfall plot. Now, does anyone know what a waterfall plot is? Hint. Okay, so you're going to see a lot of this. So this would be basically, there again, this is all sort of that CAT scan thing, which you'll grow to understand that clinical research also needs to encompass how the patient is doing. Okay, this is just the CAT scan. This is just the measurements. So that's very important. But if you just look at this, whenever you see things coming down below, that means you're seeing regression. And then this is sort of what we call stable disease, because it's really not robust enough. And then these are tumors that are growing. And you're going to see this over and over again. When you go to meetings and they say, I have the best drug, then all of it will be below. Or I have the worst drug and all of it's above. Steve talked to you about this. If anyone has questions, we can answer that. So one of the things that we're doing is really now developing mice that are more like this, more like patients in terms of what we need for immunotherapy. What's the problem with the mice that we put our human tumors in? How do they grow human tumors without attacking them? Because they're a mouse and it's a human tumor.
It's foreign.
Exactly, it's foreign, and we basically remove their immune system to get that tumor to grow. They're bred like that. So you can't test immunotherapy because they don't have an immune system.
Wow.
So that could really restrict development. So what we're doing is developing mice that we actually put human stem cells into that will populate an immune system. The reason I'm doing that is I want to be able to test immunotherapy plus something and do it in a model before I take it to a patient. Otherwise, we're going to be stuck just doing a million studies like pharma is doing. So, I mean, this is really pretty self-explanatory, but I think that, you know, one of the things that Nancy's been involved with that has been very impactful at the level of cooperative groups and research, you know, clinical research groups, has been moving faster, you know, moving forward in a patient selection way so that it's not just let's test everybody. And also making sure that the endpoints that we're looking at are relevant for the population. And I think these are things, you know, and like the oxaliplatin issue, these are things that you really can have a lot of impact on. And this is just our team. And I'll stop there. Thanks.
Thank you.